Hello, I'm Kathryn Monopoli. I'm a PhD candidate in Anastasia Kurova's lab at UMass Medical School in the RNA Therapeutics Institute, and I'm co-advised by Dimitri Korkin at WPI. And today I'm going to talk about some ways that we're working to overcome some challenges with developing therapeutic siRNAs, and in particular, a method we're developing to build models to overcome limitations in the available data. As a brief overview, small interfering RNAs, or siRNAs, are small, approximately 20 nucleotide double-stranded RNAs that can be added to cells, and once inside the cell, they can be loaded into a protein complex containing argonaute. One of the strands of the siRNA is cleaved, and the mature RNA-induced silencing complex, or RISC, is formed with a single strand of the siRNA. The RISC complex can then bind its target mRNAs through complementary base pairing. Argonaute can then cleave the mRNA, and the mRNA is degraded in the cytoplasm and the RISC is then recycled. This process is highly potent and specific, making siRNAs useful for therapeutics and research tools. Unfortunately, the efficiency of this process varies for different siRNA sequences, making it challenging to select effective siRNAs. siRNAs as therapeutics are really coming of age, with 4 siRNA drugs having been approved in the past 4 years. But I show this slide not only to present the clear utility and potential of siRNAs as therapeutics, but also because the timeline of siRNA therapeutic development and design is quite interesting. As we all are likely familiar, RNAi was discovered many years ago in 1998. And in this discovery, we learned that a perfect complementary sequence is important for silencing. Then a bit later, we discovered the importance of selecting unique sequence targets to mitigate off-target effects. We also learned the importance of thermodynamic bias and RISC loading to ensure proper strand incorporation. In early siRNA design, these were some of the major rules. Then, in 2005, the first siRNA clinical trial occurred. But in 2006, it was discovered that siRNAs can stimulate the immune response in vivo, which had a major impact on siRNA therapeutic development. It was determined that much of the therapeutic effects of the first siRNA drug were due to off-target effects. And we determined that therapeutic siRNAs have a much more strict requirement than these 3 basic rules of design. In addition, therapeutic siRNAs further face challenges with stability and delivery in vivo. And chemical modifications are kind of critical for overcoming all 3 of these challenges, including the stimulation of the immune response. So I show here an example of a chemically modified siRNA. This is the specific siRNA chemical modification pattern used in the dataset I will be presenting in this talk. So we have the 2' ribose modifications as well as backbone modifications and a 3' modification to enhance delivery. So these modifications kind of perform all of the 3 things that I mentioned on the prior slide, including evading the immune response and enhancing delivery. But in addition to that, unfortunately, these modifications also alter RISC loading, they alter mRNA targeting, and alter cleavage of the mRNA. And these all happen in a way that's really hard to predict. And that increases the challenge when it comes to siRNA design. And in fact, we've shown in a previous paper, which I have cited at the bottom here, that algorithms for native siRNA or naked siRNA design are actually inaccurate for modified siRNAs. When it comes to developing methods for modified siRNA design, we face a lot of challenges. First and foremost, there are no publicly available methods for modified siRNA design. Synthesis of modified siRNA is extremely expensive and time-consuming, and that makes developing any sort of high-throughput screening infeasible. Further, large data sets of modified siRNAs just simply don't exist in the literature, and any industry data sets are proprietary. So what we work to do is to come up with a solution, essentially a way to work with these smaller modified data sets to actually develop a model that is useful. So the dataset that we work with is a published dataset of 365 siRNAs, and you can find this in a paper we published back in 2018 to NAR. So I have the siRNA shown here. Each individual bar represents a single siRNA, and we have the target gene expression along the left side. So this dataset consists of siRNAs targeting 17 different genes, and they all have the identical modification pattern scaffold that I presented a few slides back. And lower target gene expression indicates higher efficiency of a particular siRNA. So for building our model, we use the target mRNA sequences, which we know. So this is the region that the RISC actually interacts with on the target mRNA. And we use the gene knockdown efficiencies, which are plotted along the left side as kind of our target values. So for selecting a model, we often hear that regression is kind of the obvious choice when we show people this data set, but that's simply because of the shape of the data. However, when we actually look at this region of moderately effective siRNAs, I have highlighted in red, that kind of dominate our data set, we can kind of see why this might not be the case. So for one, these moderate effective siRNAs are kind of ambiguous. We can't actually distinguish the source of their decrease in efficacy. It could be due to a decrease in RISC loading or some issues with mRNA targeting, for example. And further, these data are just not as valuable. We actually are more interested in predicting the extremes. We really want to predict efficient siRNAs or know if an siRNA is going to be very inefficient. We don't care so much about predicting the moderately efficient ones. So classification, in fact, is more suitable. And for this, we selected random forest classification due to its faster learning rate. And of the machine learning methods, it requires the fewest training examples. But one major challenge with this data set is that it's actually not obvious how we should classify our data. So one major issue is actually the data noise. So if we were to simply divide the data at 25%, which often would be what a biologist would be considering to be an inefficient cutoff, we would identify anything to the left of this as efficient siRNAs and everything to the right as inefficient. However, if we look at this classification boundary, we see a lot of overlap in the noise, and this is just simply due to the type of data. So essentially what this means is that these siRNAs that are around this boundary are indistinguishable in their efficacy. So to overcome this, we develop a trichotomous partitioning system, and this allows us to overcome this noise overlap. However, it does produce this large portion of siRNAs that are undefined, and this kind of can decrease the data available. So one way that we can overcome this is to then explore a range of threshold pairs to kind of balance this data loss. And we do this by exploring a whole range of combinations, as well as considering all cases where we have those undefined groups I presented prior. And this results in 45 different threshold pairs that we're going to consider. And I'll lay this out on the slide later on in kind of a way that will be a little bit clearer how we can see all of this. So very briefly, the sequences, so the target sequence that we're encoding, was simply encoded as a flattened feature vector of zeros and ones based off of the 4 different bases possible. And the way that we trained our models, we started, as I said, by classifying our input data, taking that continuous data and converting it into our trinary kind of classification pattern. And then we split our data into a training and holdout set. And we took that training set and performed k-fold cross-validation with it. And after kind of modifying our model parameters, we moved forward with our final model built with the entirety of the training set and evaluated on that cyan holdout set, which was never involved in any model training and therefore kind of acted as a substitute for new unseen data. And I show throughout here these simplified plots that represent how you're going to evaluate the performance. And I want to just quickly touch on that. Because of the large range of threshold pairs that we're considering here, we have a great potential for class imbalances. So for example, if we pick this 15% efficient and inefficient threshold pair, we end up in this case where we have many more inefficient siRNAs than efficient. And this can be really challenging to evaluate if you don't select the proper metrics. So for this, we use a precision recall curve because it allows us to evaluate in such a case where we have these imbalances. And specifically, we use the area under the precision recall curve, or the AUCPR. There's still challenges, though, when we consider that we're changing the positive classification threshold. So if we consider the case where we have identical positive classification thresholds, as in this example, we see that the precision recall curves all end at the same value when recall is equal to 1, and they're easily comparable. However, if we change the positive classification threshold, like in this example where we increase it to 35%, we see that the precision and recall curves look different, and the precision at recall equal to 1 differs. And this can actually lead to a phenomenon where we have identical AUCPRs, and these curves are essentially incomparable. So the way that we get around this is by adjusting the AUCPR. So the way that we do this is we take the value of precision at recall equal to 1 and we kind of subtract out the area of the curve underneath this region. And we do this for both of these curves in this example, and we can see that now the AUCPR adjusted is more comparable across these curves. And I should say that this is very specific to the type of problem we are looking at. Adjusting the AUCPR is not always the best solution to overcome this problem. We're changing the thresholds. Okay. Because we're evaluating many different possible threshold combinations, we end up with 45 different combinations that I have plotted out here, and we have them arranged so we have our efficient siRNA thresholds along the top and our inefficient siRNA thresholds along the left side. And I have our curves for the evaluation of our model on the 25% holdout set. So this is evaluation of our final model. And I have these curves colored by the normalized AUCPR that's been adjusted. And in general, what we see, and I know these curves are kind of small, but we see this general trend of overfitting as we increase the efficient threshold and underfitting as we decrease the inefficient threshold. And this kind of makes sense because we're either adding additional data or decreasing the amount of data that we're including in our models. But we find that this region of threshold cutoffs tends to be the region where most of our models perform best. And we actually identified this particular threshold cutoff of 22% efficient and 52% inefficient as our top performing threshold. We wanted to compare our random forest model to a simpler linear model. So we built a model using the same threshold pairs, using a linear method that we published in a paper in 2018. And we overlay the linear precision recall and random forest model precision recall curves. And here I removed the coloring because we're comparing across thresholds and therefore don't need the adjusted AUCPR. So if we focus in just on this top performing region and we compare the AUCPRs thusly, we see that it is clear the random forest model is outperforming the linear model, and this exemplifies that there is a benefit of our method of leveraging these data to fit a more complex machine learning model despite the small data set available. And then if we focus in more closely on our top picked threshold pair, we can see that even in this case, we have significantly greater performance of the random forest model. But now we would like to actually examine these models because we might be able to gain some biological insight into the mechanism they recapitulate. However, we run into a problem because these models are random forest models and they're kind of challenging to tease apart. So our method for extracting base preferences from our model is evaluation-based, model agnostic, and most importantly, simple to compute. So we start with our target site sequences from our mRNAs. We then convert this to a feature vector, and we compare the actual efficiency from our dataset to the model predicted efficiency, and then group each of our target sequences by their evaluation class. We then take each evaluation class group, and we compute the average weight at each base position. And we do this for all of our 4 evaluation classes. We then multiply the false positives and false negatives weights by -1 because these values represent incorrect evaluations. We then sum the weights, and we can convert this into a weight matrix. To validate our proxy feature extraction method, whose results are shown at the top, we considered a linear model where base preferences can be extracted directly from the coefficients, and I have that matrix shown at the bottom. Comparing these results with those extracted from our proxy method, we can see the preferences in general agree, validating our method. These matrices align within the base preferences of the target site of the risk complex, thusly. Therefore, we can then use them to gain some biological insight. Here I show the matrix extracted from the random forest model generated from the selected high-performing threshold pair. Examining these preferences, it is possible to identify several biological features that are consistent with what is currently understood regarding the risk mechanism, including weak base importance in the seed, which is necessary to accommodate a range of seed sequences, high affinity, shown here as greater GC preference near the cleavage site, and flexibility in the 5' region, which has been shown to aid in release of the cleavage product. When we compare this with the linear model, generated using the same threshold pairs, we see a high correlation between the two models, and in fact, the lack of preference at position 9 is shared across both models. And we know that base pairing in this region is believed to be required for efficient cleavage. Overall, however, the linear model shows lower resolution, further supporting our efforts in developing methods to apply more powerful machine learning models to such a small data set. I have shown here the GC preferences subtracted from the AU preferences for both the linear and random forest base preferences across the target site, and this can serve as a proxy of thermodynamic effects. And as you can see from this plot, we can more clearly see that the biological preferences across both models are shared, and in fact these models are quite similar. So in conclusion, we present a major challenge in siRNA design, which is the chemical modifications that limit the efficacy prediction not only biologically due to their impact on the risk targeting mechanism, but also due to the data availability being limited. By applying a trichotomous data partitioning method, we overcome some limitations in siRNA data noise and size to build a model applying machine learning methods that outperforms existing linear models previously believed to be the limit with such a small data set. We then present a simple method to examine the model feature preferences that can be applied to any classification model type. By examining our model's feature preferences, we see they do in fact recapitulate biology. And this approach of model examination can be extended not just to confirm, but to actually gain biological insight into a mechanism, which could then be tested experimentally. With that, I would like to thank the members of the Kurova and Corkin Labs and my thesis committee, as well as you all for listening, and mention that a more in-depth manuscript of this work has recently been submitted to NAR, so hopefully in the future you all will get a chance to take a look at that.